Hi, everyone. Welcome to back to the WebEVTalk series. So today is 17th August 2021 in the morning in Australia, and 16th August actually in the USA, a bit late in the afternoon. And it's my pleasure today to have Dan Lark. He's an assistant professor of health and exercise science at Colorado State University in the USA, and also director of the Extracellular Regulation of Metabolism Laboratory. So Dan Lark primary expertise is in cellular energetics and molecular physiology, and his interest in EV biology was motivated by an interest in understanding the contribution of skeletal muscle tissue to the systemic effects of EVs. Dan's laboratory seek to understand differences in EV secretion between tissues during health, disease, and aging. So today, his lecture is going to title Extracellular Vesicle Secretion, Tissue-Specificity, and the Impact of Health and Disease. So before we start, I'd like to thank our sponsor, Horiba Scientific. Thank you so much for sponsoring this session. And if you guys are interested to be our sponsor as well, please don't hesitate to contact us or go through the patreon.com and find WebEVTalk.
Well, thank you very much for the invitation, and thank you all for coming. As Carolina described, so my lab is really interested in the contribution of tissue-derived EVs to systems level function. And so I just wanted to start off by giving a shout out to Dr. Kramer Elbers from Germany, who gave a presentation about, I'd say 12 days ago on XRVs. And it was really great watching that seminar because it opened my eyes to not only the state of the art of the XRV field, but also how fast the EV field itself is moving. It seems like every other week, we think that we're doing things the best possible way, and then we turn around to find out that there's a new challenge or a new level of complexity that we hadn't appreciated. And so today what I'm going to do is share some data from a preprint. It's on bioRxives. You can see right here. And what I wanted to point out is that it's definitely a moving target for us to be able to nail down what we think is going on, especially when I talk about the fluorescent mouse model. And so I just want everybody to bear in mind, and I'll point this out as we go through, that most of the work that I'm going to talk about today was done in between 2018 and 2019, before COVID. And fortunately, COVID gave us a little bit of time and a little bit of a breather to reflect and to absorb and to really adjust. I'm not technically an EV. I don't have training in EV biology per se. We've been in this field for a couple of years now. And so I'm going to point out where we are trying to continue improving and enhancing the quality of our data as we go forward. So please bear that in mind as we go on. And so I also wanted to start by just going through the acknowledgement slide. I feel like I'm always rushed at the end, and I don't do service to these folks. So Andrea Estrada was a postdoc fellow who joined my lab when I first started. She's since moved on to a great industry position, and Zach Valenti, who's a PhD student in my lab, has really taken on that responsibility in the lab. We have a few great collaborators here at CSU. Chris Allen, who's the managing director of the Flow Core, came to appreciate how difficult it is to run flow cytometry on anything, let alone EVs. So Chris has been really instrumental in getting those experiments off the ground. Nicole Krug-Garcia is really the exosome expert here at CSU before I came. And so I learned a lot about exosomes and EVs from her. And then Tom is a colleague of mine in the department, and he and I have been working on some bioinformatics and analyzing some existing single cell data sets that I'm not going to be able to talk about today due to time. But there is another, we have another preprint. It's out, it's under review, but it's also on medRxives, looking at the tabula muris senis data set and some human single cell data sets as well, really focusing on EV biogenesis genes. So if that interests you, feel free to take a look at that. We've been funded by the American Heart, and I just wanted to again acknowledge these industry folks, because without the commercialization of these technologies that we all use, we'd really be limited. I know for certain at Colorado, a place like Colorado State, where we don't have huge amounts of core resources everywhere, it would be hard to do a lot of the work that we do without having really powerful industry folks who are able to commercialize instruments and make them available to scientists. So thank you for that. And so today I'm going to start off with an overview of EVs in health and disease. This is probably going to be a lot of stuff that you're already familiar with, given the audience. Then I'm going to go into some of our tissue-specific EV secretion data. This is ex vivo data, so these are tissue cultures. We really kind of were inspired by Sophie Rome's group back in 2014, I want to say the paper came out, where she showed that you could take skeletal muscle tissue, incubate it in cell culture media, and collect EVs from there. And so as a mouse physiologist and an animal physiologist, I was really intrigued by taking tissues as opposed to monocultured cells and studying EV secretion from there. And then to kind of piggyback off of that, really what we spent the majority of our time doing up until this point is working with a dual fluorescent reporter mouse, with the goal being to track cell type specific EVs in different compartments. Maybe that's the tissue compartment for the skeletal muscle, and maybe it's the blood. And so I'm going to show you some of the data that we have, and then also talk about a few other studies that have also looked at that and try to reconcile the findings thus far. And so when we talk about extracellular vesicles, most of the time, I say some of the time, most of the time, something like that, we're really focused on two different populations of extracellular vesicles. That would be microvesicles up here in the top right. And these are generated by budding outward from the plasma membrane. And so we generally understand the microvesicle composition to be similar to the cell of origin. Whereas exosomes are formed first through endocytosis, and then packaging and sorting in early endosomes, and then processed into multivesicular endosomes, which are then either degraded by the lysosome or released out of the cell as exosomes. And so due to this more involved packaging and processing pathway, exosomes tend to have a little bit more distinct cargo, and are also rather heterogeneous in their composition. And so we have a number of hallmarks or protein markers or molecules that we know of that are can be found in exosomes. That's certainly not to say that all of the things on this slide are found in all exosomes. That is certainly something that we've come to appreciate over the last few years, is the heterogeneity of the individual vesicle. But from an analytical standpoint, one of the increasingly popular ways of studying EVs, and exosomes in particular, are through these tetraspanin proteins like CD81, CD9, and CD63. And you can see just by this image that these have extraluminal protein domains, and these the epitopes on these proteins can be detected by antibodies. And so there's been a growing interest in using immunoaffinity, immunopurification approaches to isolate EV subpopulations based on these markers, or tagging them with fluorescent antibodies, so that way you can detect them, labeling exosomes kind of externally. So this is a very common, or it's increasingly common approach to studying EVs. And the reason why we are interested in EVs, we as in the field, is that these have been shown to be involved in a large number of biological processes. Things as general and ubiquitous as the regulation of gene transcription and translation, survival and proliferation, angiogenesis and wound healing, the kind of original, or I believe to be the original mechanism by which exosomes were supposed to be functioning, which is a waste management system. Obviously, we've come a long way since then. And then here on the right, we have just another list of really important, relevant biological processes. And because of my training and my expertise in molecular physiology, cellular energetics, metabolic disease, we're really interested in the metabolic reprogramming and regulation. And we're not just interested, so these arrows pointing away from the exosome sort of suggest that these are the things that exosomes are involved in, and that is true. We're also interested in what the metabolic events that are occurring in the cell are doing to exosomes. So exosome secretion, exosome composition. And so we're really thinking about this bidirectionally. And so in the context of metabolic disease, really, oftentimes this starts as a hyperglycemia, insulin resistance, but these are some of the more common metabolic abnormalities, I guess you could say, in Western society. So abdominal obesity is another one. These are meant to depict adipocytes. And these converge on what we call the metabolic syndrome. This is really a constellation of metabolic diseases that contribute to poor health. So these are all comorbidities. And again, these interact, so you can see how hypertension, elevated circulating fats, abdominal obesity is a good example where abdominal obesity can manifest in some folks to be insulin resistance, elevated circulating fats, have some crosstalk with the adipose tissue itself. So there's a lot of interaction between these different conditions and between each other. And so we're really interested in skeletal muscle as a tissue because skeletal muscle is influenced by these different components. And so, for example, there's some really nice, elegant work from, I'd say, I think 20 years ago, probably now, showing that elevated circulating fats by themselves can cause insulin resistance in the skeletal muscle, abdominal obesity through a number of mechanisms can affect the skeletal muscle, and the skeletal muscle is the primary site of glucose disposal, and therefore, when the skeletal muscle becomes insulin resistant, as it can and often does, this can have serious implications for metabolic health. Now, not only is skeletal muscle the primary site of glucose disposal after a meal, but it is also the organ or tissue that is most arguably most active during exercise. So we contract our muscle to perform exercise. And we know from a large body of literature over decades that exercise improves metabolic health, and it does so in a number of different ways. It improves circulating fatty acids, it reduces insulin resistance instantaneously. A single acute bout of exercise can eliminate insulin resistance in a number of populations. And when combined with diet, exercise can improve adiposity or reduce abdominal obesity. And so it's this, again, this bidirectional sort of interaction between skeletal muscle and metabolic, these metabolic parameters that we're really interested in. And so for the sake of today's talk, I really just want to emphasize as well that our interest in adipose tissue alongside muscle is really that each of those tissues are the two largest in the body by mass, skeletal muscle and adipose tissue. And so we were really interested in understanding EVs being secreted from those tissues. And then it'll make a little bit more sense in just a minute. So this is from a recent review article, just showing that there's published work showing that EVs from non-vascular cells, and what I mean by that are cells that don't directly interface with the vasculature, that the cells that secrete those EVs can then have effects in autocrine, paracrine, and sometimes endocrine mechanisms. Okay, and so here in the purple are depicted as skeletal muscle EVs. And we can see that skeletal muscle EVs can act back on the skeletal muscle itself, presumably through an autocrine or paracrine mechanism, to affect gene expression, namely the cell cycle and differentiation. However, there's also evidence to indicate that skeletal muscle-derived EVs can affect the pancreas. and alter microRNAs and signaling pathways in the pancreas. And the only way for that to occur, since the skeletal muscle does not exist in the same tissue as the pancreas, is through endocrine signaling. And so in order for this to occur, skeletal muscle EVs in vivo would have to be able to get into a biofluid, whether that be blood plasma or the lymphatics. And so there's very little evidence that EVs from non-vascular cells, or there's not very little, I should rephrase that. There's limited evidence. It's hard to draw firm conclusions on the number and the distribution of in vivo secreted EVs from non-vascular cells. So it's unclear when the skeletal muscle fiber, for example, secretes an EV, where that EV is going and what it is doing. This is not a trivial question. Certainly, this is something that is has been discussed in the field for years. This is not unique to us, our interests, or anything like that. But suffice it to say that we know that these many different non-vascular cells, like skeletal muscle myofibers and adipocytes, secrete EVs. We know that from monoculture experiments. And so we're really interested in pushing that forward and getting to a point where we can understand the in vivo biodistribution and hopefully ultimately function of non-vascular cell EVs. And so in the setting of type 2 diabetes, at least, and overnutrition in rodents, there's evidence that EV concentration goes up. Now I have to be careful here, and I can thank Dr. Kramer-Albers for this because I learned this from her talk the other day, is that lipoproteins are highly abundant in human plasma and mouse plasma. We know that, but what her group has found is that lipoproteins are co-isolating with EVs even using size exclusion chromatography. And so these studies were done using centrifugation. The studies I'm about to show you were done using centrifugation. But just know, at least the evidence is moving in that direction, that it is incredibly challenging to get pure EVs from plasma. And so, but in the context of type 2 diabetes, this was a paper came out of Michelle Evans' group at NIH. They found that individuals with diagnosed type 2 diabetes had more particles per mil, and presumably some of these are EVs, maybe some of them are lipoproteins, are higher than individuals that are euglycemic or without diagnosed type 2 diabetes. There is a paper in PNAS that came out about the same time in mice that showed that 15 weeks on a high-fat diet also tended to increase the concentration of particles per mil compared to a chow-fed mouse. And again, this is in the plasma. And so the early indications, I think it would be reasonable to presume that EV concentration increases with high-fat feeding. And so that might be indicative of some sort of signaling mechanism. And so then what about exercise? So we talked about metabolic disease and the factors involved there. Exercise is a very sticky area right now. It's very challenging for a number of reasons. One is really just, again, quantifying the EVs in a biofluid. And the other is trying to figure out whether skeletal muscle is contributing to the circulating EV pool. And so this was work that was done published back in 2015 by Dr. Kramer-Albers' group, where she had, by their own recognition, a pilot study where they had a few subjects where they performed a cycle ergometer test, starting at 50 watts and then increasing an incremental cycle ergometer test. And what they found was that particle counts in the plasma increased post-exercise relative to pre-exercise. This was true in both of the subjects shown here. And that they had similar but less compelling evidence on a treadmill test. Now, there is a growing discussion that the intensity of the exercise is really important, particularly exercise that is sufficient to cause damage to the tissue? And that presents a really interesting question of what is the role of the vasculature in regulating EV trafficking. It's something I'm not going to touch on, but I just wanted to throw that out there because I think it's a really, really interesting question in and of itself. And so if we skip forward a few years to Martin Whittam and Mark Fabreo's paper in Cell Metabolism, where they did their own cycle ergometer tests, collected plasma, and then measured particles again, and they saw the same thing. And so this, again, gave some early evidence that exercise could increase circulating EV abundance. However, as she recently described in her WebEVTalk 2 weeks ago, they went on, Dr. Kramer-Elber's group went on to generate a larger cohort of subjects, perform more studies, controlled the physiological variables much more tightly. What they found was, in contrast to their original paper, they did not see an increase in particles in the plasma when they isolated EVs with size exclusion chromatography instead of ultracentrifugation. And so this, and this is just the individual data points for the different groups to show you that there was no difference. And so this creates this conflict right in the field where there's some evidence that it increases and some that it doesn't. And really what we want to, one of the big gaps that we have, right, just kind of added to the list is EV clearance. So what is EV clearance in vivo of in vivo secreted EVs, the circulating EVs? That's something that we still don't have a good handle on unless I missed something recent. And this is just a YouTube link to Dr. Kramer-Elber's talk. Like I said, I really enjoyed it, and it was a nice primer for this. And so I showed you some data in the plasma, but what's going on in the tissues? And so this was a paper from Sophie Rome's group back in 2014, where she collected the vastus muscle of the mouse. So this is like the quadricep muscle of your leg. And she cut it into pieces, or her team cut it into pieces, placed it in cell culture media, and then incubated it for 24 hours, collected the conditioned media, and then isolated EVs. I believe this was done using ultracentrifugation. And what they found was that the skeletal muscle secreted more EVs in high-fat fed mice than in chow-fed mice. And this was, again, I think 15 weeks or so of high-fat feeding. This was a chronic high-fat diet. So then Flaherty et al. In 2019 in Science looked at adipose tissue EV secretion, and instead of doing, and did actually, sorry, a very similar experiment where they compared lean and obese mice. I believe these were OB mice. Don't think these were high-fat diet fed, but I might be wrong on that. And showed essentially the same thing, where the exosome EV release rate was increased in the setting of obesity or overnutrition. And so the kind of the overall consensus, at least thus far, is that both exercise and high-fat feeding may increase EV secretion, and that this is appears to at least be occurring at the level of the skeletal muscle and the adipose tissue. Now, where we stepped in was really being interested in going back to, I guess, first principles, which would be, if you have two tissues that account for a large quantity of body mass, and they're both capable of secreting EVs, and they're both flexible in their ability to secrete EVs, then we should probably want to know what's the capacity for these tissues to secrete EVs just under normal conditions, just relatively speaking. And so our first question really was, do skeletal muscle and adipose tissue secrete a similar quantity of EVs? And then the second question is, does contraction contribute to skeletal muscle tissue EV secretion? So because we know that one of the obvious differences between skeletal muscle and adipose tissue is the ability of muscle to contract. And we know that this happens under experimental conditions and can be an artifact. And so we did some experiments to control for that. And then finally, can we track muscle fiber-derived EVs in vivo using these fluorescent reporter mice? So can we go from tissue EV secretion in a dish to circulating EVs in the blood? So we started this journey using other people's data, namely the Human Protein Atlas. And this is just to give you a sense of what resources we used for this. So we took the normal tissue data, which is immunohistochemistry of tissues. We had RNA tissue consensus, which is individual cell types within a tissue. And then we applied gene ontology, and namely the extracellular vesicle biogenesis gene ontology. We added a few genes here, so I will be honest here, this is not just the gene ontology for extracellular vesicle biogenesis, which is really aimed at exosome biogenesis and secretion. So it incorporates a lot of the escort complexes, ALEX, and those components, where I added two in particular, these ARR DC1 and 4, which are implicated in microvesicle secretion as well. And so just to orient you to this table, we have adipose tissue here, it's skeletal muscle here. This is again IHC data, immunohistochemistry data first. And these were categorically defined. So 0 meant that the protein was not detectable with IHC. And then 1, 2, and 3 correspond to low, medium, and high expression. And all we did here on the right side is compare these two numbers and figure out which one was bigger. And if it was larger in skeletal muscle, then we put skeletal muscle here. And so you can kind of scroll through this and see that there were 9 proteins that were higher in expression with IHC and skeletal muscle than adipose. There were 12 that were equal and none that were higher in white adipose tissue. So this gave us a pretty interesting early taste of what to maybe expect with our EV secretion experiments, where it appears, at least at first glance, that skeletal muscle tissue has more of the machinery needed to produce EVs. And then if we look at the RNA sequencing data, now we're looking at a continuous variable, so we don't have anything that's equal. It's either skeletal muscle or adipose. And you can scroll down here and see that there's 13 that were higher in skeletal muscle, 10 that were higher in white adipose, and 6 of these genes were higher in skeletal muscle in both the IHC and the RNA. Those 2 here at the top, again, are involved in microvesicle secretion. And we haven't yet gone into parsing this out, but this is just, again, to show you that based on the Human Protein Atlas, that there is some indication that skeletal muscle might be capable of secreting more EVs than adipose tissue. And so the way we've been measuring EVs up until now has been nanoparticle track and analysis, and this has been using a Horiba Manta ViewSizer 3000. This was an instrument that was originally developed by Manta, and then Horiba has since taken on and commercialized and distributed the product, as I sort of indicated at the beginning of the talk. And really the value in the uniqueness of the ViewSizer is in its laser capabilities. And so a conventional NTA has 1 laser, and it's typically in the 400 nanometer range. And the reason why is because you can see in this blue box that in order to detect a small particle, you need to have a low wavelength of light. And so that allows small particles to scatter more light. However, large particles scatter lots of light. And this can impede the ability of the user to detect small particles in a polydispersed sample. So if you have particles of varying sizes, the large particles will kind of block out, in a sense, the small particles. And so the way that the ViewSizer works is that it uses 3 separate lasers, and it has software, and the software will pick and choose which laser to use to interrogate a given particle. So a large particle would be examined using the red laser, because the red laser would not saturate the particle the way that the blue laser would. So if you had a large particle and you only have a blue laser, you run into some interference of that large particle in the detection of small particles. So the ViewSizer is able to pick and choose which laser to use to interrogate your particles. And the other, I mean, I'm sure there are a number of unique aspects of the ViewSizer, but the other one that I think is noteworthy from an application standpoint is that it's a cuvette-based system. So typically with NTA, we have syringes and we have pumps, and we pump at a given flow rate, and then we fill the window, and then we analyze in the window. Well, this is a cuvette-based system, and it has an insert where this circle is, and this is a black insert, and it allows the instrument to detect particles continuously, and it has a star bar in the bottom, and so it mixes the sample and distributes all the EVs equally, and then it'll take a video when there's no flow, and then it'll stir and then take another video, and so on. And so it's nice in the sense that it's one sample in a cuvette, and it's constantly being mixed, and so it gives you a nice representative output of your particle concentration. I do want to note, if there are any heavy cuvette users in the audience, that I did build a very easy DIY cuvette dryer. So one of the big challenges with the ViewSizer, I will admit, and it's also a challenge with other NTA instruments, just in different ways, is that if your glass is not clean, it is really hard, I would argue impossible, to get good quality data. And so we've developed a workflow in which we clean the cuvettes with water ethanol, and then we let them dry for 5 minutes. And it's a really nice, clean, consistent process. It really keeps costs down, because we don't have to have 5 cuvettes in a rotation in order to do experiments. We can really get away with 2. So anyways, if you're interested in building one of those, like I said, it's very simple. It uses a pipette box, which you probably have many empty pipette boxes these days with the supply chain issues with COVID, and it's just a little PC fan, and that's it. So anyways, if you're interested in that, please let me know. And so we used the ViewSizer to measure EVs secreted from the skeletal muscle. And so we did this again from the vastus medialis, which is part of that vastus complex, the quadricep, and we used epididymal adipose. Specifically, this was the perigonadal adipose tissue fat pad. We incubated those tissues and we cut them into small pieces. We aimed for 5 to 10 milligrams per piece. We incubated them in DMEM with pen strep for 24 hours, 5% CO2. We then centrifuged at 3,000 Gs to remove cell debris, and then isolated with ExoQuick. Now, I will say that ExoQuick, I'll show you a little bit of the data later, that precipitation has not, it was a good place for us to start because it was pretty straightforward, but we're definitely expanding our capabilities as far as our EV isolation is concerned. And so here's the data. And if you look on the left side here, we have particles per mil, and this is normalized to the milligrams of tissue in the well. And so this is important to note, because we ended up having to use, we only needed to use 50 to 100 milligrams of the muscle to get a good quality signal, but we needed to use 500 to 600 milligrams of adipose to get enough EVs to isolate to do NTA. And so when you do the math and you normalize this data, this ends up being about 100-fold difference in EV concentration. You can see that this is on a log scale. And each one of these lines is an individual mouse. So we're connecting these. This is a paired analysis. And so this is a pretty pronounced effect size to be detecting. We didn't see any differences in mean or modal diameter from these experiments. And so my background in mitochondrial energetics really kind of led me to this next experiment where when I was a master's and a PhD student, I learned from a good friend of mine, Chris Perry, that that skeletal muscle in a dish when you prepare it for mitochondrial studies, we use what's called the permeabilized fiber technique, is that skeletal muscle will spontaneously contract in a dish. And so that we weren't intentionally looking at contraction per se, but we understood that it was a potential artifact. And we were worried that spontaneous contraction could increase skeletal muscle EV secretion based on the exercise data that's out there, and that might explain why the skeletal muscle had such a high EV secretion rate compared to the adipose tissue. And so we used blebbistatin in the experiment. So we just did the same experiment. We just had wells with blebbistatin, wells without blebbistatin. And we saw no difference in EV concentration. And so we were a little surprised at this, but we kept on pushing forward. And so the next question we had was, well, the other big difference between muscle and adipose, besides the fact the muscle contracts, is that their metabolic phenotype is very different. Skeletal muscle is very oxidative. It has a fair quantity of mitochondria in it. Adipose tissue does not white adipose tissue at least. And so we thought, well, if we took two different muscle types, one that was more oxidative and one that was more glycolytic, would we see a difference in EV secretion? Specifically, would the soleus have a greater rate of EV secretion than the plantaris? And so we did this experiment, and indeed we did find that the soleus secreted more EVs than the plantaris. Now, we weren't the first ones to find this. And so I put the citation down here at the bottom. This is from Tim Gavin's group at Purdue University, and they actually found this in 2019 in their paper in Experimental Physiology. And they used the enzyme assay, acetylcholinesterase, and found the same thing. And so this is comforting in the sense that at least two different labs with different techniques came to the same conclusion. And so, so that was good to see. So if you wanted to see more about that, you can take a look at that paper. And so, but, you know, as we can appreciate now, precipitation of EVs is not ideal. It's often not sufficient for getting a pure clean EV prep. These are exocheck blots. So these are dot blots of different EV markers. GM130 is a cytosolic marker. It is a contaminant. It's not supposed to be there, and yet it is. In both the skeletal muscle and the adipose tissue. This is probably a consequence of us chopping up the tissue and putting it into a dish, right? And so some of this may be unavoidable, but at the same time, we can do it better, and we are currently. So right now, we are in the process. We've isolated most of the samples using size exclusion. We're going to repeat the NTA experiments, or we're in the process of repeating the NTA, and we're also able to obtain a TRPS instrument, the new Exoid instrument from Izon. And so we're going to repeat all of these analyses with EVs that are isolated with size exclusion using NTA and TRPS. So more to come on that. Hopefully we'll be able to get all of this into the paper that's in revision. And so just to summarize so far, muscle certainly appears to secrete more EVs than white adipose tissue, at least in mice. We're definitely interested in doing the same set of experiments in humans. The differences cannot be explained by spontaneous contraction. I'm not showing you the data today because I don't have all the methodological details. We do have a collaborator at East Carolina University, Dr. Espen Spangenberg, who he and his group performed a number of electrical stimulated skeletal muscle experiments and also found that there was no increase in EV abundance with electrical stimulation. And so that, again, that data would have been great to have, and I just wasn't able to put it together in time. But there's growing body of evidence that skeletal muscle from our group, and then also from the folks who have been doing all these exercise studies, that there's a lot still to be learned, I guess, about what skeletal muscle as a tissue is actually doing for EV abundance and secretion and systemic function. And then finally, that skeletal muscle tissue EV secretion at least appears to be determined, at least in part, by what I phrased intrinsic metabolic capacity, but maybe it would be better described as metabolic phenotype. So oxidative versus glycolytic muscle has different rates of EV secretion. Why that is, we don't know. Okay, so do EVs reach the circulation in vivo? Skeletal muscle EVs reach the circulation in vivo. Well, we developed a dual fluorescent reporter mouse to study this. And the rationale behind this was really that EVs are everywhere, and we don't really have great markers. We have alpha-sarcoglycan as a putative marker in the skeletal muscle, but it hasn't been able to be reproduced across all labs, which, I mean, very few things in science are, but certainly it begs the question of what more do we need to know? And so we were fortunate that the American Heart Association gave us a grant when I first started the lab to pursue this idea, and certainly we weren't the only ones doing it then, and we're certainly not the only ones doing it now. And so what we used was the Mtmg mouse. The Mtmg mouse has been available for, I think, over a decade now. It's been used for cell lineage tracing and countless studies. And really the way that it's designed is it has Cre-mediated expression of either a membrane-targeted TD tomato, which is a red fluorescent protein, or a membrane-targeted enhanced GFP, which is a green fluorescent protein. And so in cells that don't express Cre, we have expression of TD tomato. And the hypothesis that we had was that the cells that lacked Cre would secrete EVs that contained membrane-targeted TD tomato. And in cells that contained Cre, and we designed a mouse that specifically expressed Cre in skeletal muscle myocytes, that those EVs would contain enhanced GFP. So we'd be able to color code the EVs based on where the Cre recombinase was expressed. So other people have used this mouse model to study EVs. And so this is a paper from Claire Crew when she was working with Phil Shearer at UT Southwest. And what they did was they took adipose tissue from an MT mouse, and so the red fluorescent protein expressing mouse, and transplanted it into a halo mouse. And so this was a mouse with a different, gene engineered profile where they had a green fluorescent protein, and they were just looking to see when you transplant a red fluorescent protein expressing cell into the green fluorescent expressing mouse, do you get mixing? So do they turn yellow, right? Because it's a combination of red and green. So this is the microscopy from that paper. And you can see that proximal to the implant, so not the red fluorescent protein expressing adipose tissue, you see red fluorescence, indicating that this TD tomato has somehow made it proximal to the implant. You can see the nice halo expression here, and then you can merge and you can see some little bit of overlap. Now, if you look at where the implant is, you can see that there's lots of red fluorescent protein, halo, and much more of this mixing. And so this indicates that these TD tomato labeled that, for one, that TD tomato expressing cells secrete EVs that have TD tomato, and that these are taken up by naive cells. And so these appear to be functional EVs. Now, another paper this is from Heather Pua at Vanderbilt University, generated two different mouse models with two different Cres, one for the lung epithelium and one for hematopoietic cells. And here she was looking at flow cytometry. So here we have TD tomato on the x-axis, and then side scatter, which is just indicating where your particles are. And you can see that both mouse, and this is from lung alveolar lavage fluid here, sorry. So this is the biofluid that we're looking at as alveolar lavage fluid. And you can see that there are TD tomato expressing particles in both of these samples. And if you look for GFP now, and so again, these lung epithelial cells have GFP in this model, and the hematopoietic cells have GFP in this model, you can see that the lung epithelial cells, because they interface with the lung, with the alveolar lavage fluid, that you accumulate these GFP positive particles. And this is just quantified here, showing that in these lung epithelial cells that are interfaced with the lung alveolar fluid have GFP positive particles, whereas MT mice that don't have Cre, and even MG mice that have the hematopoietic stem cell Cre, have less than the lung epithelial cells. So this sort of indicated that at least in the local tissue environment, that these GFP positive cells can secrete GFP positive particles. Okay, so then what did we do? We took our mice, and these are now, we did a number of studies with MT mice and these skeletal muscle MG mice. So again, our CRE is targeting skeletal muscle fibers. We took skeletal muscle as a positive control. We wanted to see, okay, if we took tissue, we put it into a dish and we harvested the EVs, we should see some GFP. If those EVs are present, we should not see them in the adipose tissue because the adipose tissue does not express CRE and therefore should not express GFP. And then we collected blood. We did the exact same isolation extraction technique. We used our view sizer for NTA, and then we used an Aurora spectral flow cytometer for our flow experiments. This is not a nano flow cytometer. We came to appreciate how nice having one of those would have been, but alas, here we are. And so what we found from the skeletal muscle, this is first in MT mice. So these are mice that don't have CRE, everything is red. So what I'm going to do is I'm going to show you some flow data here. And on the y-axis is going to be your side scatter, just like we saw in the PUA paper. And then on the x-axis is going to be TD tomato signal. And so with PBS alone, you see some scatter, but you don't have any EVs in there, so there's not much. If we put wild type EVs in, these are from wild type mice, no fluorescence. We see lots of scatter, but no fluorescence, just as we would expect. But then we add MT EVs, and we see the profile completely changes. And so this isn't to say, I should be clear that this experiments that we've done that I'm going to show you, in large part were designed to be binary. They were designed to be proof of principle. Is it there or is it not there? And so certainly there is a lot more that we could and should do as far as the quality of our flow data and the clarity of our flow data. But again, we were learning, we started from nothing. And so we're certainly picking it up as we go. And so if we quantify these events, you can see in wild type mice, there's no TD tomato positive events because there's no fluorescent protein. And these are skeletal muscle. And when we do the same experiment in the MT, just as I just showed you, nearly all of the events are TD tomato positive. When we do the same experiment in adipose tissue, we get about the same response. And so we strangely have a little bit of fluorescence here. This is really driven by one mouse. This is probably artifact, maybe carryover from a previous experiment. We're not sure. And we have relatively less MT, but the trend is the same. There's clearly MT positive particles being secreted by adipose tissue in these MT mice. And so then what about GFP? So we did the same experiment now with MT mice, again, isolated with ExoQuick, and then ran them on the flow cytometer. In this experiment, we started with a bead mixture. So this is the apogee bead mixture. And we have here a number of populations of unlabeled beads. These are a mixture of latex and silica. And then we have GFP labeled 110 nanometer and then 500 nanometer. I believe these were latex particles. So diffractive indexes, very different, fully recognizing that our ability to detect small EVs using this instrument was certainly limited, without a doubt. So what we did was we started with this bead mixture and then we spiked in EVs from our MG mice. So these are skeletal muscle-derived EVs from the MG mice. And you can see that we populate the space where the GFP is and where our particles are. And so again, this is not a quantitative experiment. This is not what you would do if you were trying to estimate the number of particles of a given population. This was really, again, just a proof of principle to show that when we put green EVs in, they came out where we would expect them to come out. So I'm just going to show you a couple more individual experiments, then I'm going to wrap up here. Again, this is just wild type EVs now versus MG. GFP is on your Y, tomatoes on the X. You can see in the blue here, there's no fluorescence in the wild type EVs, whereas you go to the MG EVs and you get this vertical distribution, which is indicative of a GFP signal. There's lots more details on the flow experiments too. I know that flow is very involved and complicated, and so I'm sorry I couldn't get into all the details, but they are in the preprint. Certainly if you have questions, please feel free to email me. So we did the same experiment with adipose tissue EVs. Remember, the adipose tissue shouldn't have Cre, and so it shouldn't have GFP positive EVs. And so you can see in the red here are MT adipose tissue derived EVs, and in the blue are Mg adipose tissue derived EVs. And you can see these kind of share the same space. We went on to do a detergent lysis experiment. So the question here was if there was free GFP that was co-occurring with other particles, that might be explaining why we detect the GFP, but we used MP40 here. So we either ran the experiment with MP40 or without MP40, and you can see that without MP40, we have GFP signal. Sorry, the axes are switched around here on you. X-axis is eGFP. But when we add the detergent, we lose all detectable fluorescence, indicating that the eGFP was localized to a particle and probably most likely to some degree undetectable at the single fluorophore level or fluorescent protein. And so just an interesting anecdote here, we're still trying to reconcile this, is that when we looked at MT mice in the skeletal muscle, 99% or so of the events were MT positive. When we did the same experiment in MG mice, only 50% of the events were MG positive, and less than 10% of those were MT positive. So we lost. So we went from almost 100% fluorescence to only 60% of our events being fluorescent. And so again, this question of signal to noise and the detection level that you can get at, we know with this instrument that we are working at the level of the noise. We're right up against it. And that has always been a challenge when we've been doing these experiments. But what happened to that other 40% of fluorescent events? Well, the short answer is we don't know. But one guess that we have is that the brightness of eGFP is only about a third of TD tomato. And so if you have small particles that maybe only have a few copies of Mg, of the green fluorescent protein, you may not be able to detect that with the flow cytometer given the capabilities of the instrument. So just something to ponder there. I just thought that was interesting and throw it in there. And now for the really disappointing part of the talk, at least it certainly was when we did this, is looking at the plasma. And so here we have TD tomato on the X, GFP on the Y. I'm going way late, I'm sorry guys. And so here we have our two quadrants. So these are MT positive, these are MG positive. This is wild type plasma. We shouldn't see anything. We see very little fluorescence in either quadrant. When we do the same experiment with MT plasma, we see some MT positive events. Certainly it's not to the same level as we did, as we saw with our tissue culture. And that is probably in part because there are so many lipoproteins and other particles in the plasma that would look like EVs, I guess, on flow. And then finally, in our MG mice. Now this is really where we thought we were gonna have a slam dunk and really be kind of cruising. And it turns out there is very little up in this MG quadrant. We get maybe a tiny bit of a change here, but again, this is an N of 1. This was just a couple of representative experiments, and we kind of hit pause after this because we were stumped. And so the big challenge here has been how do you find these EVs? And so we stopped for a while, and we were pretty frustrated and disappointed, and we didn't know where to go next. But then we started seeing all this data on the immunocapture and thought, well, maybe this could work. So, the next question, I'm just going to show you one little last bit of data on immunocapture to see if we can detect these endogenous EVs. And just to point out, I'm just going to breeze through this, is that Alexandra Brommer's group, her paper with the Kramer-Albers lab, showed that when you pull out these subpopulations of EVs, CD9, CD63, CD81 EVs, and you compare them pre-post exercise, you get changes in some of these tetraspanin proteins, which you might expect if you're getting EVs into the blood. But there was no detection of this alpha-sarcoglycan, which is thought to be a skeletal muscle specific EV marker. And so that's not to say that it isn't, it's just to say that they didn't detect it. And so the jury is still out, I guess, based on this work on the extent to which skeletal muscle-derived EVs get into the blood with exercise. Certainly, we don't see them in the plasma with the flow cytometer. So anyways, I'm going to breeze through this this was just to show that Heather Pua's group also did not find any fluorescent EVs in the very few fluorescent EVs in the plasma with the Mtmg mice. So plasma EVs have been really, really hard to study with endogenous fluorescence, presumably because of all the other co-isolated factors. And so I mentioned these tetraspanins and how we can use antibodies to detect them and grab them. And so recently we've been working with NanoView Biosciences. They have a really cool platform that I think I describe it as an ELISA with a microscope, where it's an immunocapture assay, and then they apply fluorescent antibodies on the top end. So it starts off like this. A conventional NanoView experiment would be a solid phase with CD9 antibodies or CD63 or CD81 antibodies adhered to this microfluidics chip. And then you would incubate your EVs on this chip. And any particle that bound up to the antibody would be indicated of having CD9 protein. And then you would place a fluorescent antibody on top of that. So this is here where it kind of looks like an ELISA, right? And then you would be able to say, okay, well, my particles that have CD9 also express CD63 or CD81. So it's a really nice tool to first immunocapture and then directly detect. Now, for us, we actually have, we can simplify this to only 2 steps, because instead of having to use a fluorescent antibody, we can just put our EVs directly on there and let the endogenous fluorescence do the work. And so what I'm about to show you is some skeletal muscle EVs from Mtmg mice that were isolated with size exclusion chromatography. So these should be a nice clean prep. This is an image of the data that we're working with. This was some preliminary experiments that they ran for us. We have a whole cohort of studies that are currently going. Just to orient you, red is the CD63 antibody that I showed you. So this is a CD9 capture. So every dot on this screen is a CD9 positive particle. The red is CD63, the green is the TD tomato, and the blue is the endogenous GFP. I'm sorry for the change in the colors here. And if you zoom in, you can see these individual particles with these individual colors, and some of them have some mixtures. But we can really nicely see very small particles with these different colors on them. And so we're really, really excited about this. And so 57, in this experiment, 57% of the CD9 captured particles were positive for MT. Roughly 20% were positive for MG. And so this gives us, I think, a pretty high confidence output of these. And the reason why I have such high confidence in this data is that instead of working at the level of the noise like we were with the flow cytometer, we're actually getting about the same fluorescence intensity from our endogenous proteins that we are from the CD63 antibody. So that makes us feel pretty good that we're not really low on the signal to noise, and that we're risking getting false positives. There's always the risk and there's always the possibility. We always have to be careful, but this is very promising data. So I'm going to summarize. Muscle and adipose both secrete endogenously fluorescent EVs or particles ex vivo. We couldn't find very many muscle fiber derived EVs at all in the plasma using flow. We are going to run a bunch of plasma samples on the NanoView platform and that same immunocapture assay that I just showed you. So we're really looking forward to that. That will give us a much clearer picture of what's going on in the blood, we think. And so stay tuned for that. Future directions, we have some samples that we have mice that we fed a high-fat diet. So we're going to look to see what's going on with the muscle fiber derived EVs in the tissue and in the blood. Really kind of have a big picture question of how do non-vascular cell-derived EVs cross the vascular endothelium? That is an open-ended question that I think is really important. we think about these things in the context of tumors, and tumors have a perforated, leaky endothelium, and so they're not really comparable to a healthy skeletal muscle or a healthy adipose tissue. Whereas if you have mechanical disruptions of the vasculature where you have a leaky vasculature, then that's obviously a different story. And then I mentioned the cell population study earlier, and you're welcome to look at that as well. Thank you for your time, and I'm sorry for going so long. That was certainly not my intention. Thank you.
Okay, thank you so much, Dan. Wonderful presentation. Let's stop the screen share first. We actually a little bit over time, so I think we have just a few minutes for question and answer. If anybody wants to ask question, please go ahead. Michael?
Can I just dive in? First of all, Dan, hi, nice to meet you. I'm Mike Rayner. I'm down the road from you at CU Anschutz. And so I'm pretty familiar with Nicole's work, for instance. So along with all the introductions and stuff. So there's several things. One, what about the, say, uptake by the liver of the vesicles that get spit out of the muscle? And secondly, what if they're contracting? Would you see more during, say, an exercised mouse, as you were seeing in humans? Would that then have them show up in the bloodstream more effectively than what you're doing?
Yeah, so the exercise studies are definitely on our list. We had some breeding issues that were kind of a secondary consequence of COVID. We had to shrink our colony, and then our mice decided to stop breeding, of course. And so we've been waiting to do those studies. Really good question. So about the liver, I've always been intrigued, and I guess I have to be careful that I'm not being naive here, but my understanding is that the studies that have been done that show liver uptake are generally, if not entirely, based on exogenously delivered EVs. And there's in my mind, there's a question: if you're going to have non-vascular cell-derived EVs in the blood, how are they getting there, and are they being modified at all? And so, the question of, they all end up in the liver, which I think you could reasonably expect that if you overloaded the body with EVs, certainly the liver would want to clear them out. But what would be, the kind of conventional in vivo biodistribution? You wouldn't want to, I think teleologically, you wouldn't want to dump a bunch of really energetically expensive material into the blood just to have it all get taken up by the liver, right? And so I don't know the answer to that question, but it's something that we've talked about. We've thought about, just pulling the liver from these mice, and even just doing like a Western for GFP. And we have some frozen livers that we could do that with. We just haven't gotten around to it yet.
And you're encountering the issue with sensitivity, that we all know that most of these fluorescent probes just in vivo are really difficult to track out. If you could like radio label your mice, then you'd probably be able to do this, but not so sure how much they would like that.
Yeah.
Rad mice running around. So
we have some ideas on some isotope experiments, but yeah, doing it all in vivo, would be really challenging. I think, that's why I think the mouse models that have the cream-mediated fluorescence are really interesting. If we could just get the signal up. And I'm hoping that the immunocapture gives us that sensitivity, and at least at single particle level. So I feel like if we're gonna find something in the plasma as far as muscle-derived EVs, that's how we're gonna find them.
Right. We've been trying to obtain one of those exoviews from NanoView for a couple of years now, working through a funding mechanism at our university, and we've basically been told to kiss off. So hopefully somebody will come up with one of those one of these days.
Yeah, we just have a CRO with them right now, so we just contracted out some samples, and they've been great to work with so far, though.
Yeah, they are good. They're Clayton Dagan is very good.
Yeah, that's who we're working with.
Right. I'll shut up.
Well,
thank you, Michael. Thank you, Dan. Just have a sort of a question for the NanoView data. Actually, was that actually the tissue, or is that the EV from circulation in plasma?
So that is, those were skeletal muscle tissue-derived EVs that were isolated with size exclusion chromatography. We also analyzed them with the ExoQuick, and we got similar results. They were a little different, but looked comparable in many ways. But no, so we haven't done the plasma yet. What we're really excited about is that we don't have to or we've been told that we don't have to purify the plasma to do these experiments.
Yeah, that's correct. I actually use that for plasma, and it's
Yeah.
pretty good. Yeah.
Awesome. So that's good to hear. So we're just going to send them just straight plasma from chow and high-fat fed animals and see what we get back. We have a couple of controls too, some MTs and some wild types to just kind of check all the boxes, right? But yeah.
I think there was a talk in the past, forgot who gave the talk, but there was shown of the mitochondria in the EV itself. It was a scanning electron microscopy image. So I think that probably something that can be interesting to look whether you also see mitochondria inside the EV.
Yeah, I've been fascinated by that. I think I need to take a little bit more time to really dive into it. I was a little skeptical at first because I'm like, well, the lumen's not large enough to fit, entire organelles and things, but maybe. But no, it's been on our mind, but we don't have a dedicated study to that yet.
All right. Thanks so much, Dan. Again, wonderful presentation, and thank you for sharing your data with us, and really good luck getting over the COVID and get your study running.